Hello, everyone, and thank you for joining us today. I'm Sean Fensky, Editor-in-Chief of Orthopedic Design and Technology and moderator for today's program. You've logged on to the ODT webinar, Switching Sterilization, What Should You Consider When Changing Processes? Before we get started, I have a couple of housekeeping announcements. For those of you who are interested in the information you hear today, once the webinar has ended, or if you'd like to forward it to a colleague, this session will be archived for a year. The archive will be able to be accessed through the ODT website, which is at odtmag.com. Also, we will be accepting questions throughout the presentation. You can type your question into the box provided in the webcast window. We'll collect all questions and hold them until the end, but please feel free to type in questions as you have them. And now, before I introduce today's speakers, I'd like to take this opportunity to thank our sponsor, Nelson Labs. Nelson Labs is a global leader in microbiological and analytical chemistry testing and advisory services for the medical device and pharmaceutical industries. The company serves over 3,500 customers across 15 facilities in the United States, Mexico, Asia, and Europe. Nelson has a comprehensive array of over 800 laboratory tests supporting customers from initial product development and sterilization validation through regulatory approval and ongoing product testing for sterility, safety, and quality assurance. And now let's get started. Today we're joined by three members of the Nelson Labs team. Bryce Telford is a recognized authority and industry leader on radiation sterilization, bioburden, microbiology, and tissue processing. He has over 13 years of experience working in laboratory functions in research, test design, media formulation, medical device, and tissue industries. Jennifer Giggy has over 28 years of medical device laboratory experience. She has worked in the microbiology, bioburden, IDs, and packaging and validation sections at Nelson Labs and has experience with various microbial tests. Audrey Turley has over 25 years of experience working in research, laboratory, and test design function in the medical device industry. She is a biocompatibility expert, having spent 15 years in the lab performing all the in vitro tests offered at Nelson Labs. Now we'll begin with today's presentation. Bryce?
Hello, everybody. Welcome. So today we're going to be talking about considerations for changes in sterilization modality. So I'll be starting things off. So today's learning objectives, we're going to talk about some general considerations when it comes to changing sterilization modalities. Then we're going to dive into some case studies of actual changes and considerations that go with them. So we're going to start with EO to radiation. That's one of the most common that we're seeing in the industry right now, and therefore we're going to make sure that's the first case study so that we discuss that topic. Also, EO to EO. So with that, that's mostly to do with cycle optimization. And then we're going to talk radiation to radiation. So that's going to be your changes such as your gamma to E-beam. Now, there's a lot of different iterations that we can do with these. We're going to focus for our case studies on the more common topics. And then finally, we're going to finish up with EO to alternative gases, such as vaporized hydrogen peroxide and the noxalizer. So to begin, we have the overkill methods. So we generally break sterilization down into two categories. So first is overkill, and for that we're going to use a most resistant organism. Commonly in the industry we call that an MRO for short, and that's going to be used to create a biological indicator, again BI for short. And we're going to use that also to sometimes create what is known as a process challenge device, so a PCD. So quite a few differences there where we're going to be using a most resistant organism, a biological indicator, usually with up to 1 million CFU on it, and we're going to use product challenge devices. So with that, your sterilization is going to generally be chamber and cycle specific. So anytime you're making a change, that's going to be something that needs to be considered. So some examples of overkill methods are your EO, steam, dry heat, and your vaporized hydrogen peroxide. The other sterilization modality is going to be your bioburden based. So that is going to be based on the bioburden of the product. There's not a most resistant organism. There's not a biological indicator. It's the actual bioburden on or in your product. And since it is based on the product's bioburden, we are going to be testing that product. And generally speaking, that's going to be a destructive test so that we can find that bioburden. So again, there's no biological indicator, there's no PCD, we're testing directly to the product. So examples of bioburden-based sterilization, that's going to be your gamma, your X-ray, and your electron beam, E-beam for short. So with that, we also are going to have situations where we're going to be changing, and how does that change in dose affect things? So in general, gamma is gamma, and E-beam is E-beam, and X-ray is X-ray. So changing from one of those to another, such as one gamma radiator to another, that's generally not going to have a large effect product and what you need to do. You would need to consider and do a new dose map with your radiator, but in general, you're not looking at a revalidation or things like that. With these methods, whenever we're talking about sterility and sterilization, what we're really talking about is our sterility assurance level. So that's mostly going to be a 10 to the minus 6 sterility assurance level or better. The other thing to consider whenever we're talking about sterility, and it's why I'm joined by my associates today, is there is no sterility without a sterile barrier system. We can go through all the steps we want to, we can sterilize it. There's not a sufficient sterile barrier system, then it's not going to matter the second we pull it out of whatever's sterilizing it. It's no longer going to be sterile. So let's jump into our sterilization modalities and go over some basics with them. The first is EO. And when we talk about EO, the general variables that we're going to consider are temperature, gas concentration, relative humidity, and time. Those are the main variables that we always consider, but there's also some additional parameters and considerations. So when we're talking about cycle optimization, we might be changing temperature to improve gas reactivity. We're also going to potentially be looking at changing vacuum cycles. So pulling deeper vacuums, having more vacuum cycles in order to improve that gas diffusion. We're also going to potentially consider product configuration. Maybe I'm going to change up how much product's in a load so that the gas can again better diffuse through the product. And also my packaging configuration. And this is going to include secondary and additional packaging, doing things such as adding chimneys or changing the way it's on the pallet, so that, again, I can improve that gas permeation into the product for sterilization. So one of the things with EO to consider is that it can be sterilized in pallet form. Entire pallets can be loaded up and in, but with that, there also tends to be dunnage. So dunnage is where you add additional product, dummy product, simulated product, however you want to call it. Dunnage is essentially the term, where you're filling up the load so that you have the proper load configuration, the proper densities in order to meet your validation parameters. So sometimes in order to consider some of the changes, improve again our gas permeation, maybe we're going to eliminate dunnage. With EO, we also need to consider the residuals. So with EO gas, I am using a gas for sterilization, and I have to get that gas back out, and it is a pretty nasty gas. It is a known carcinogen, so we need to get those EO residuals out and account for them. And again, the main thing that we're focusing on and considering when it comes to ethylene oxide really comes down to gas penetration and diffusion. So how can I change the cycle? What can I do for the cycle in order to improve those parameters? And where you're really going to save if you do something like an EO optimization is probably on your preconditioning. if we can save any time on preconditioning or on aeration, then we can do that. But that's also a consideration with EO versus other sterilization modalities, is there is that preconditioning step. Radiation, for example, there's not a preconditioning. That's not something to be concerned with. So those are variables that we want to consider and that we're going to look at when we talk about sterilization for EO. So the next is going to be general considerations for radiations. So radiation, our variable is dose. So we don't have temperature, time, gas concentration, all that goes away with radiation, and our variable is dose. But with radiation, one thing that we also want to consider with that dose is going to be the dose rate, and we'll go over that in a few moments. But with radiation, a concern is material compatibility. With ethylene oxide and other things, it's either compatible or it's not. I can run it through the chamber or I can't. Steam, it's going to pass or it's going to melt. With radiation, materials compatibility is going to be our main concern. And that's going to change depending on how much dose the product is receiving. In general, the more dose, the more detriment there is to the product and its packaging system. So we want to consider what that dose effect is and how is that dose effect going to be affected by the materials. We want to talk about the modality. So that's our dose rate. So that's one of our big differences when it comes to gamma versus E-beam. So that modality difference, that dose rate difference is going to be that gamma is generally going to be over the course of hours in order to receive a full sterilization dose, whereas electron beam, we're dosing in minutes to even seconds. So anytime we're doing sterilization, we're essentially adding energy into the system. For ethylene oxide, that's chemical energy. For radiation, that's ionizing radiation. And anytime that we're putting energy into the system, that's going to affect product. And if we're doing it for gamma, and that's over the course of hours, and we're doing it for E-beam, and that's over the course of minutes and seconds, that's going to be a difference with that energy input. So with that, there's also some consideration for temperature. Since I am loading quite a lot of energy into the system in a short period of time, there is potential for temperature increase with E-beam. Because it is over minutes or seconds, there really isn't an opportunity for that temperature to leave. That's not going to be the case for all products, but that is a consideration for some products, is that temperature effect due to that difference in dose rate. So validations, also we need to consider that when we do ethylene oxide, as an example, we're validating a chamber and a cycle. With radiation, we are validating per product. So that dose is going to be based on what amount of radiation is needed to sterilize that product, not for that chamber setup, not for that cycle parameter. So with that, we're going to account for those cycles and those parameters with our dose map. The other nice thing about radiation is there's no residuals, there's no diffusion as such. So whatever goes in, it's not going to be any more radioactive or anything when it comes out. And as such, we don't have to worry about off-gassing, and we also don't have to worry with radiation about preconditioning. So with that, one other consideration, and this is one of the reasons one of my associates is joining us today, is E-beam needs some consideration because it is not able to handle density as well as gamma, for example. That doesn't mean it's not a better method or it's a comparable method. It just means that there needs to be additional consideration. So with E-beam, to account for that density, it's very important that you have some additional consideration for your packaging. We want to present a very similar target for that product.
All right, well, let's look at the packaging in a little bit more depth then. So what are some considerations that you would be thinking about when you're talking about your packaging? Because again, as Bryce has said, there isn't any sterility claim without having a good sterile barrier. And really, you can't have a good sterile barrier unless you have a sterile barrier validation to back that up. I've got an example here. This is a typical sterile barrier system. We have a pouch, a device in the pouch. The pouch goes into a carton box. Multiple carton boxes go into a corrugated shipper. When we're looking at making changes to the packaging system, and again, that's anything in this picture, not just your pouch, right? If you're doing that, you've got to have good change control procedures in place. And those change control procedures need to look at the holistic picture, not just the sterile barrier, but also the protective packaging. Look at the device. And maybe you're changing sterilization, as we're talking about here. How does changing sterilization affect the packaging system? Maybe you're making a manufacturing change and you might not think that would affect the packaging, but it can. So we're going to go into a little bit more depth with these examples of types of things that you would want to think of in regards to packaging and how that change may affect, so that you can incorporate that into your change assessment for sterilization.
And now we want to take a minute to think about our general considerations for biocompatibility. So as Bryce mentioned, sometimes we forget about considering biocompatibility when we talk about sterilization changes. So I put a clip in here from 10993 Part 1, which is our biocompatibility main guidance standard. And it does specify that if you change your sterilization, you do have to consider that impact on your biocompatibility. So I just want to highlight it is a relevant component of the discussion today. And when we look at biocompatibility, Jen, if you want to go to the next slide, there's a couple main categories that we look at. So first, and I'm going to repeat this throughout our presentation today, is the impact on the material. So this is a large component of biocompatibility because we are assessing materials and processes. So even though sterilization is a change in the process, that change can impact the material. So we're going to talk about impact to materials for all of these changes, and then also concerns with residuals for the various modalities.
So let's jump into our first case study, which is going to be a transition from EO, ethylene oxide, to radiation. So this is actually a very big change because we're changing from an overkill-based method where we're using MROs and biological indicators to a bioburden-based sterilization method. So one of the biggest implications is control of the bioburden. Is this being manufactured in an ISO clean room? Are the raw materials being controlled? Are there controls in place for personnel? All those things, controlled environment, raw materials, in order to ensure that we have a low or at least controlled bioburden. Generally speaking, the more bioburden you have, the more radiation the product is going to need in order to be considered sterile. With that, that leads into our material compatibility. Product and packaging need to be able to withstand that maximum dose. Radiation is going to have a different effect on the product and its packaging system than ethylene oxide. Ethylene oxide has considerations that are going to be different than radiation as well. So we need to make sure that we're accounting for those materials and their compatibility. Another thing is drug products and their activity. Since EO or steam or other sterilization modalities, generally what goes in is going to come out. Radiation, we're going to have a materials effect. And again, that I can't stress it enough, that is going to be dependent on dose. The more dose, generally the more material effect you're going to see. So another thing to consider is you're going to be changing how you're doing your releases. So for EO, you're doing batch release testing, you're using biological indicators. For radiation sterilization, you're moving to what is known as dosimetric release with routine dose audits. And those dose audits need to be done either every 3 months or per batch if production is infrequent. So some things to consider with this change is just what some of the limitations with radiation sterilization are. So the absolute lowest sterilization dose that we can reasonably achieve when following the ISO standard is around 8.5 kilogray, and that's using a method 2B validation. For those that aren't familiar, a method 2B validation is very involved, it's very expensive, so to get down to that truly low dose is actually quite a process. So if your product isn't able to be materials compatible at a dose past 8.5 kilogray, chances are radiation sterilization isn't going to be a good option for you. That's basically the floor of where we can get down to. And then for the other validation methods, such as VD Max, the lowest we're really going to get is 15 kilogray. There is a new AMI TIR that has been published, so that's AMI TIR 76. It is an AMI document, and the lowest that can get to is 11 kilogray. Method 1, the lowest dose we're going to get also is 11 kilogray. And some consideration with all of this is different modalities generally are going to have different dose ranges that the irradiator can hit. So just because I can get a low dose, such as 8.5 kilogray, that doesn't mean that's what my product is going to see. I'm going to see a range of doses. For gamma, the general rule of thumb tends to be a 1.6 multiplier. So an example of that is 25 to 40 kilogray. My product is going to see a minimum sterilization dose of 25 kilogray to be considered sterile, but it's potentially going to see upwards of 40 kilogray. For E-beam, I am generally going to see, and a general trend is what is known as a 2x multiplier. So product is going to be dosed in a range of 25 to 50 kilogray. Now, these are generalities. Things are going to vary based on your irradiation provider, as well as your product and configuration, but that gives you kind of a general idea of some of the ranges we're seeing for sterilization.
All right, let's look at really packaging validation for a second. I have a diagram here that shows you kind of a little flow chart of what a packaging validation would look like. And I think it's kind of helpful to review for a moment. So you're going to start out with your final product. You're going to run worst case sterilization, and then you're going to do a distribution simulation in order to rough up the packages and see what kind of damage is going to happen in more simulated real world conditions. Then you're going to do baseline testing for strength, integrity, and microbial barrier, and make sure that somewhere along the line with sterilization or distribution, you didn't get any holes or breaches or integrity problems with your packages. And then you're going to age the product to the end of its expected shelf life and repeat that testing again. If you are switching modalities from EO to radiation, how does that really affect this validation? Well, again, radiation is all about materials compatibility. And if the materials compatibility of the packaging that you're choosing is kind of on the fence, you may be weakening that material to the point where it may break. You may get holes or tears in the package that you wouldn't have seen in EO. And so you're really going to have to repeat this packaging validation. For sure, you're going to need to go through shipping and baseline testing to make sure that it can get through that. You may or may not have to repeat the entire validation. There is a possibility that you could find some data, maybe from your pouch manufacturer or someone, that shows what happens to that packaging over time after exposure to worst case radiation. And you may be able to supplement your validation file with that. Otherwise, you would have to, again, show that as the product and materials age after sterilization, that the packaging is still going to be robust enough to provide that sterile barrier. And this, of course, is assuming that you haven't made any changes to your packaging system, which you may or may not choose to do. And if you do, that's a whole nother conversation that we could have.
All right, so now we're going to look at our biocompatibility considerations when we switch from EO to radiation. So I've just kind of made this flow chart that we'll go through for each of these case studies. So always checking first material compatibility. I put this Amy TIR document here as a good reference. Of course, there are others, but this is, the one that I would go to first. What we want to understand is when we see that the material is compatible, we still need to understand what's actually happening to the material. So with radiation, it's likely we're seeing either chain scission or cross-linking. So with chain scission, I just use a little mnemonic trick, like scission looks like scissors. So it's that the polymer or material chains are actually cut. So you decrease your molecular weight and your strength, but you also then increase your possible leachables, which is what we care about in biocompatibility, because those studies are based on what's leaching out of the product or device to the patient. If cross-linking occurs, you can have a stiffening or an increasing in strength, so we get more of the polymers or molecules crossing together, making a denser matrix, so we don't have as many leachables. So I put an option in here if we see and we know cross-linking occurs with these materials. One thing we can do is to write a risk assessment to justify using the previous biocompatibility. So this is an example of how you could still use your previous biocompatibility without repeating it. One thing we have to consider too is oxidation occurring. So with gamma radiation, especially when we use cobalt, so this is not with E-beam, but with cobalt, we have a lot of oxygen available. So this caps the end of our chain scissions. So we don't see any likely repairs in those materials. So we can see an impact from that as well. One thing to note, if we do not know this information on the materials, on the specific materials, then repeating biocompatibility is a likely scenario. So again, switching these modalities impacts more than just the sterilization cycle.
So for our next case study, we're going to talk about a change from an EO to an EO. So an example of this is going to be our cycle optimization. So the question that comes up with this is why? If I have a sterilization validation in place, why change your EO cycle that you already have? You've done all the work. Why am I going to do another validation? Well, the first answer to that is the FDA's asked us to. The FDA's asked to reduce EO consumption and use. This is better for the environment and it's more sustainable. The other is EO residuals. So kind of the basic formula is less EO in means less EO out. So where that's really going to potentially save some time is going to be your aeration. If I'm not putting as much in, or if I'm doing different cycles and I'm using vacuum more to get it out or to get it in the first place, then potentially I can save on some aeration. Again, kind of a general less EO in, less EO out, so I can save some there. But things to consider with this, so one is going to be cycle changes itself. So we're going to be changing our gas concentration. We're potentially going to be increasing our temperature for increased reactivity of the gas. That increase in temperature, that can affect product as well as its packaging system. We're going to be potentially increasing the vacuum. So maybe we're pulling more vacuums in a shorter period of time or pulling deeper vacuums. Also looking at the loads, so our min and max loading, maybe optimizing that so that we don't have to sit in the chamber as long because we've improved how much is available. Also with that, maybe I shrink my load so I don't have to use as much gas, so I have more gas available for reactivity. I'm also going to potentially look at how is my monitoring performed. So I have a process challenge device, I have a PCD, that tends to be a worst case setup. Well, have I made it too worst case, so much so that it's no longer representative? Also adjusting my biological indicator, so the newer BI bioburden based, so I adjust my BI to be more reflective of my product bioburden. as well as liquid inoculum. It tends to be a little bit better carrier. Product packaging and configuration we're talking about are protective packaging. So maybe changing my packaging, making it so that I have a larger surface area of, say, Tyvek, something that's permeable, so that I can have improved gas penetration in the product, improved diffusion. Same thing with my sterile barrier, my wraps. I'm going to change them so that I can make it easier for the gas to get into and out of my product. So I'm going to open up pathways in the device so that it works better.
All right. You know, this was actually such a big topic that I recently wrote an article about it last year, I believe, and I put a link to that below. It's called EO Optimization and Potential Effects on Sterile Barrier Packaging. You feel free to look that up to get some more in-depth discussion on this topic. But what I've got up here is I've categorized the packaging, the changes that you would make as part of one of those optimization cycles, and then categorized them as low risk or high risk for packaging specifically. If you're lowering the gas concentration, that's really going to have less of an effect because you have less of that nasty gas to deal with. If you're going to change your PCD, that means that you're going to have a shorter cycle. That's not really going to mean you have to repeat a packaging validation. Same thing if you're going to change from the overkill to a BI bioburden or a cycle calculation, as well as if you're going to change from the paper carrier to the liquid inoculum, because again, that's just going to shorten your cycle. If you want to remove your paper IFUs because cellulose materials absorb EO, or you want to change your load configuration in the chamber to change how you've put things on the pallet or how you're loading it in there to make it so that there's more pathways for gas to get into the center of the load. Or if you're going to pull the stretch wrap off and change it to banding so that you are, again, removing a barrier, none of these things are going to impact your packaging per se. So these are all things that you could easily justify in your change control just using a paper justification. The high risk column here, these are things that are most likely going to require you to do at least some supportive testing to show that you whatever those changes are not going to have a negative effect on the packaging system. The big one here is increasing the vacuum pulses or increasing the vacuum draw. That is going to put stress on the packaging seals, and there is a potential there that additional stress might cause a seal rupture that you hadn't seen before. When you reduce your protective packaging, either by pulling out some of the cardboard, or maybe you use thinner materials or remove extra layers in your protective packaging, this is going to make sterilization easier, but that's less protection for your sterile barrier system. And so you'll need to assess whether it needs that extra protection or if removing that would be okay. And then if you change your device to disassemble it or something so that it's got more open space and that the gas can penetrate through it easier, that may change how it bounces around during distribution inside your sterile barrier. When you increase the temperature, this one's not quite as critical as some of the other ones, but you need to make sure that the increase in temperature isn't going to have a negative effect on the materials that are used for your packaging. And when you increase the breathable surface area in your sterile barrier, you have made a change to that sterile barrier package. And any time you make a change to your sterile barrier package, that's almost a guarantee that you're going to have to do additional validation work. And then think back to the diagram that we showed on the last one. You're definitely going to have to get through the top end of that flow chart and get through baseline testing to show what kind of effect this may or may not have. But you may be able to use some previous aging data to leverage in your justification to not repeat that part of the validation.
Now to move on and look at our biocompatibility considerations. Our checklist here is again looking at material compatibility. So assuming that has given us the green light to move forward, then our next concern is, looking at the residuals. So when we are optimizing or changing our cycles, things we have to look at that could impact our residuals are possibly a shorter aeration, a higher concentration of gas, or even a higher temperature. So from a biocompatibility perspective, the EO and ethylene chloride residuals that will occur from the exposure to the sterilization cycle can produce irritation responses. And we do see cytotoxic effects, which could indicate that we would see another different, maybe systemic adverse response from the patient, depending on what the product is. So, Jen's given some scenarios for packaging that is low risk, but also some of her discussion where, it's a higher risk as you change that packaging. You might also be able to work with your packaging and think about, how could we reduce that residuals by having a more breathable packaging and kind of going through this process. So there's ways to have packaging help biocompatibility, which helps optimize your EO cycle as well.
So our next consideration is a change from radiation to radiation, but not just gamma to gamma. We're going to do a little bit more of an extreme change, such as gamma to E-beam. So this is a change in the dose rate. So gamma, again, it's a low dose rate. Generally, you're receiving the dose over the course of hours. Whereas for E-beam, again, it's over the course of minutes. That's a pretty big change in how much energy is being entered into the system at a time. So that is going to potentially reduce the oxidation and improve materials compatibility for the product. However, that's really not the only consideration that we need to have from a materials compatibility standpoint. So the ISO standard does give some guidance that a product qualified at a low dose rate, such as gamma or X-ray, will typically require minimum qualification to demonstrate material compatibility at a high dose rate. However, that's considering all things being equal. So just because the dose rate difference doesn't mean I'm not going to see a difference also in my dose range, again, that we demonstrated earlier. And that increase in dose is going to affect materials compatibility, and we need to be mindful of that. From a sterilization perspective, we also need to be mindful of that density. So E-beam is not able to penetrate materials as well as gamma is. So with that, some very dense materials aren't going to be a very good option. But mostly that's going to mean a reconsideration of how I'm packaging and handling my product. So with that, I need to present a consistent target to the E-beam. So we don't want loose product in pouch. For gamma, I might have something that is basically in a pouch. It's kind of loose. For E-beam, that loose material is potentially changing how it's being presented to the E-beam. So we kind of tend to like something like an organized tray. The other thing is we are going to need a new dose map. So with that dose map, as we mentioned, we're going to be susceptible to changes due to that density. So things such as adding IFUs, thickening the IFU, changing our carton density, that's all things that are potentially going to affect an E-beam and how well it can sterilize the product, as well as your dose range. So changes that we're going to need to consider with sterilization are going to be that density. The other thing is, whenever we make a change from gamma to E-beam, or really any of the modalities, we really need to demonstrate that the microbial effectiveness is still there. So we probably don't need to do a whole new validation, but we generally are going to be looking at and needing to do something such as a verification dose experiment, which generally we're going to handle by doing a new dose audit to show that change in modality is still able to effectively kill all of the bioburden that we need to in order to achieve sterility.
All right, so back to packaging. Let's look at this change. The big thing that we need to talk about is when you're going with E-beam, and Bryce has hit on this, you need to have a consistent presentation to the source, because the density, the penetration of E-beam is not as good as with gamma. We need to make sure that we one, reduce the density as much as possible, and two, that it's always going to be the exact same. The problem with the spaghetti tubing here in my picture on the left is that density in one area is really low, but it's really high in another area. And if I have loose packages inside of a box, they may shift around. And again, I don't have consistent density throughout the box as it's being presented to this source. The picture example on the right is a much better one where the tubing is coiled, and then you would, so that it's all monolayer, then you would also need something to keep that in place, whether it's a tray or you have some kind of a clip insert that holds it all in place, as long as you can keep it all uniform so that every time it goes under the E-beam source, the E-beam is getting exactly the same product every time. And then the other consideration we need to look at here is really, did the max dose change? Because essentially, you know, dose is dose, and that effect on the material is based on dose. And if my max dose has increased, then the material's compatibility has most likely decreased. And I have more change scission, and I have more cross-linking. I'm going to get more brittleness. I'm going to get more rigidity, less elasticity in my polymers. This is going to make them more prone to, again, cracking and tearing and developing holes. And the other thing that may, even if your sterilization dose hasn't changed, your max dose may change because of that multiplier. You saw in Bryce's example that for gamma it was a 25 to 40, whereas that could go up to a 25 to 50 for E-beam. And anytime that the max dose changes, you're going to have to do some testing to support that. Again, you can probably on this one just get by with baseline up to baseline testing to show that a little bit higher sterilization dose is not going to negatively affect the packaging in such a way that it will develop sterile barrier breaches during use.
So for biocompatibility, again, we're checking our material compatibility, and we're staying focused on dose, just like Jen and Bryce have already covered. So as Bryce said, you know, if you keep your dose the same, so our hazard symbol here is gamma in this table, and then I have the electron symbol for our E-beam. So if it stays the same, then you can usually do a justification here where we don't see a lot of concern from a biocompatibility perspective. But if the dose increases, just as Bryce was saying, we see an increased impact on the material. So if you're seeing cross-linking with gamma at 40, you see even more of it with E-beam at 60 kilogray. So that does change our leachability of the profile of the material, so it needs some additional consideration. And also want to think about temperature sensitive products, and that can be impacted from a biocomp perspective because it'll heat up a little bit more in E-beam because it happens so fast that the heat doesn't have time to dissipate from the impact of the rays, or of the beam. So we have to think about, does that release even more compounds or more leachables to the surface of the device that then would be exposed to the patient? So one thing I want to highlight here is that it becomes increasingly difficult in this case to do like a literature review and say, oh, you know, this material has a history of this with E-beam, to say it's exactly the same with the material of the device, unless it's a consistent material and uniform across the device. So as we get into some of these nuances of these changes, we are looking at reperforming biocompatibility testing because it's just, we need the data to make a conclusion on patient safety for the change.
So our next and final case study is going to be when we're changing from EO to an alternative gas. So that's a broad category. We're going to kind of lump in vaporized hydrogen peroxide and the noxalizer, for example. Big considerations here is that each one of these is going to be its own validation. So the STERAD, there's different machines, there's different cycles, same with vPro. So just like EO is cycle and chamber dependent, very much the same case when we're using those alternative gases. The other thing is those alternatives really haven't been set up on an industrial scale. So there's not really industrial sterilizers that are made to use large amounts of vaporized hydrogen peroxide or the noxalizer. There are some situations where the noxalizer is able to do, for example, large pallets, but this isn't going to be something where you're going to send an entire semi truck and it's going to be done. The Sterad and the V Pro are generally about the size of refrigerators, so you're going to be doing some lower quantities. Also, with that, you need to be very mindful of how you're doing your validation and how it works. So the Sterad and the V Pro, for example, you need to make sure your product fits within the 510K submission. So a common example would be something such as a flexible endoscope. You have a long and somewhat small lumen that may not fit into the parameters that are set up for the 510K for something such as, say, an express cycle. So you also have to make sure that your product is able to fit within the limitations set by that 510K submission. You are going from an overkill to an overkill method, so that is going to be very similar compared to, say, a change to radiation, but you are potentially changing your most resistant organism. And you also need to be considerate of where the packaging and sterilization is going to occur. A lot of times, if you're switching from EO to an alternative gas, you're switching from an industrial sterilizer and your packaging to something that is probably going to be packaged by the end user, and it's going to be sterilized by that end user.
All right. That's an interesting thought with these alternative methods of sterilization, is that you just don't have the volume capacity that you would with EO or with radiation. And so that puts you in a bit of a quandary. If you're doing low batch manufacturing, it would be possible for you to, just sterilize these one box at a time, essentially. There's a bit more materials compatibility to look at. Some of these alternate gases are not as compatible as EO with certain material types, so you may be using different types of packaging. Another option would be to sell your product non-sterile with the expectation that it will be sterilized in the hospital setting. If you're doing that, that doesn't necessarily absolve you of running a packaging validation. So I would suggest, if that was the way you were going, you'd think about the customer impact, because hospitals prefer to use sterile devices rather than sterilizing their own. They do have a lot of these units available to them in the hospital, and they can use them to package and sterilize items. Generally, these, though, are not single-use items. They're usually reusables. And you'll see types of packaging, such as these pictures I have below. This is just a little medical-grade paper film pouch. It's not an adhesive seal, or it's not a heat seal pouch. It's an adhesive seal. And those are more problematic because you don't have any control over the closure of your packaging. And part of the packaging validation is really validating that you can close this sterile barrier and make sure that it's consistent every time. And you lose that when you're having someone else or many someone else's package the product. Another type of common sterile barrier system that's used in a hospital is surgical wrap. And that's my middle picture here. And again, this depends on, the person assembling this and folding this. This is not exactly uniform every time. And then there's the sterilization containers. And these are usually used for surgical kits. Again, all reusable devices that have been processed and cleaned, generally speaking. So if you were going to go this route and you were going to produce this in a large amount and you were going to sell it as non-sterile, you should consider how the hospital is going to package it for sterilization on site and make sure that it's compatible, not just with the type of sterilization as a VHP or an oxalizer, but to make sure that the packaging that they would select was compatible, and that sterile barrier system that they're likely to use is going to be able to handle the sterilization cycle, because the hospital isn't going to do a validation to make sure that that sterilization cycle will work with your product. So you would really want to cover that yourself.
For biocompatibility for VHP and Noxalizer, what we're really looking at, especially for VHP, is going to be any residual hydrogen peroxide that remains. So I know that, you know, the residual for the sterilization cycle is stated to be water, which is true. But what we see is that there are still some residuals of hydrogen peroxide that are low enough to actually cause a positive response in either the cytotoxicity test or the irritation test. So these are tests we run for biocompatibility. So what we would recommend is if there is a switch to VHP, that there would be some residual hydrogen peroxide testing performed. Because then with that, we can understand, even if it would show up positive in the biocompatibility tests, if we run a residual test and can quantify the amount, then we can do like a toxicological risk assessment to understand if there would be an adverse effect to the patient based on how the device is used in the exposure and the patient populations. We could take that into consideration. For Noxalizer sterilization, you know, the one thing we know is the gas is incredibly toxic. However, we know also that it's a great method for sterilization because it doesn't leave concerning residuals. I think the biggest issue, which is what Jen talked about, is material compatibility. So I just popped in this table here from the standard TIR 17 that shows, you know, what materials are incompatible. And if you see, a lot of those are very common materials in medical devices: nitinol, polyurethane, nylon. And so I think it's a limited window of materials or devices that could go to this Noxalizer sterilization. But if you can, and it works with, you know, the other conditions that Jen was talking about, it seems to be a good alternate if it meets the needs. So that covers the last component of our case studies today. So just to wrap up, I wanted to put our contact information for those of us that presented to you today. And also, if you find you have other needs for medical device support, you can certainly reach out to our advisory services at Nelson Labs. And this goes to our team that Jen, Bryce, and I are all a part of. And we can at least get you in contact with the right person if it doesn't happen to be one of us. And then I also wanted to highlight something that we have available on our website, which is our Sotera Health Academy. And this is an opportunity for you to search our content that we have available, which is webinars, white papers, and other things that relate to more of your questions or med devices. And so you can go to the link that's here and gather information on content and then search for yourself and do a little bit of self-training or obtain training for your team. So other than that, just thank you for joining us today.
Fantastic. Well, thank you to all three of you for that thorough and informative presentation. Now it's time for the Q&A session. We'll try to get to as many questions as possible. For all three of you, I'm not sure who wants to take this, but the first question, with the cycle change for an optimized EO cycle, how does the change affect the original 510K submitted for the product? Will new 510Ks be required for each product, or is FDA offering a quicker path when it comes to the original 510K submitted?
So that is a good question, one that we don't have a direct answer to at the moment, but the EO cycle optimization and the EO changes are something that regulatory bodies, particularly the FDA, it is a directive. So they are looking at ways to make it a faster process and improve the process. We believe that that's probably more of an amendment to your 510K than an entirely new 510K. But that's something that our ethylene oxide experts with our advisory services can certainly guide you through and gives you some additional guidance on and how best to navigate that.
Fantastic. Thank you. The next question we have, what can I do if my product has high bioburden but is radiation sensitive?
So That's something that comes up when we're transitioning from, say, ethylene oxide to radiation, is we didn't have the same level of control with the ethylene oxide because it was an overkill method. So now we're wanting to switch to radiation, but we find the product's radiation sensitive. And there are things that we can do. The first, of course, is what can we do to get the bioburden down? So are there processing steps, disinfection steps, something like that that we can add? Of course, if we're adding disinfection steps, then we have residuals, and we're also going to need to consider our biocompatibility. But there are things that we can do to improve our materials compatibility. So some examples of that would be dosing on, say, ice or dry ice, eliminating oxygen from the environment, such as doing a foil pouch with a nitrogen purge or oxygen absorbers. So there's also some things that we can do to improve the materials compatibility a bit. The other is considering our sterilization method. Again, method 2B, that's the lowest dose that we can get. That's very set to the what's known as D value of your product. So the amount and resistance of your product's bioburden, and that'll help us get basically the dose down in your sterilization. So hopefully that we can handle the product.
All right, fantastic. The next question. Are there products that are not suitable for E-beam?
So that is going to depend on the E-beam irradiator that you're using. But in general, if you're kind of doing materials that are not going to be radiation compatible, they're definitely not going to be E-beam. Well, I shouldn't say definitely, but
No crystal ball.
there's no crystal ball, and E-beam does tend to be a little bit better on materials compatibility, but as a general rule, if you're looking at TIR 17 and it's not compatible, it's probably not going to be a great option for E-beam either. But also products that are dense, so very dense, so large pieces of metal, things that are also high density, such as large amounts of liquids or things like that, they're not going to do as well with E-beam. So that's going to be something that you would need to either work with your E-beam provider to see if there's changes to the configuration. Maybe you're just doing one product at a time or something in order to get it through the E-beam. But it also might be a case where you're working with your E-beam provider and the dose uniformity just isn't there, so you have too large of a range, and that's putting you out of your compatibility. So it's kind of a case of, yes, there's sort of, kind of, it depends, depending on the irradiation provider and your product.
Okay, I think we have time for one last question. Are packaging suppliers properly gearing up for new different materials, in particular for EO to gamma or E-beam?
You know, that is a very, very good question that unfortunately we don't have the answer to because we're a testing facility and we don't know what manufacturers are doing. I would hope that they have seen this on the horizon because this has been going. The FDA issued their challenge in 2019. Obviously, COVID put a brake, a big slowdown on a lot of things, but it's been out there for a while, and our sterilization company, sister company, Sterigenics, has been working on a lot of these optimized cycles with clients. And so the ball is rolling, it's gaining steam and momentum, and I hope that they're getting prepared for it, but it's hard to say because we don't have visibility to that. And I'm sure that every manufacturer is experiencing some of the same supply chain issues that we have experienced as well. So that may come into play.
Okay, appreciate that. But unfortunately, that is all the time we have for today's presentation. I'd like to thank our presenters, Bryce Telford, Jennifer Giggy, and Audrey Turley for their insights. I'd also like to thank our sponsor, Nelson Labs, for its support. Finally, I'd like to thank you, the viewer, for attending this session. I hope you found it to be a valuable experience.